Bismillahirrahmanirrahim. Let's talk more about the chlorinated aliphatic hydrocarbons when they are used as electron acceptors. Now, as we discussed before, there can be different reactions which can be involved in the conversion. And the conversion of dichloroethene to vinyl chloride and vinyl chloride to ethene, it is much slower as compared to the initial steps where perchloroethylene was converted to trichloroethene. Why so? They have different thermodynamics, they have different structures, and different enzymes are involved. Depending on that, the rates will be different. Now, the hydrogen that serves as electron donor, it is normally produced by the fermentation of different organic compounds. If we talk about benzoate and propionate and their fermentation, you can see here that the fermentation of both of these compounds basically leads to the production of 3 moles of hydrogen each. However, in case of benzoate, it is producing 3 moles of acetate, which is 2 more as compared to propionate. So therefore, if you talk about percentage of electrons converted to hydrogen, it is much higher in case of propionate, about 43%, as compared to benzoate, which is 20%. Now, the hydrogen which is released by this fermentation can be used by different types of microorganisms present at that site. Those microorganisms can be sulfur reducers as well, iron reducers, manganese reducers, as well as methanogens. And if you talk about methanogens, those can use hydrogen and in order to grow. However, in order to utilize hydrogen to grow, the methanogens need a certain threshold concentration of the hydrogen. If the hydrogen is present below that concentration, the methanogens will not be able to use that hydrogen to grow. Now, if we talk about how much hydrogen should be there for the methanogens so they can use it, the threshold level is about 10⁻⁻⁶ atmospheric pressure. If we translate it in the form of concentration, it is mostly 8 to 10 nanomoles. The threshold for dehalogenation is much lower, which is about 1 to 2 nanomole only. So you can see here, if the methanogens are present as well, they will like to use the hydrogen which is just generated due to fermentation of organic matter. And however, the methanogens cannot use hydrogen until the concentration of hydrogen is at certain level, which is, you can see here, 8 to 10 nanomole. However, the other microorganisms present in the same environment at the same site, and the microbes can cause dehalogenation of the compounds. Those also need hydrogen as electron donor. However, they need much more low concentration of hydrogen, which is about 1 to 2 nanomole only. The thresholds for sulfur reducers, iron and manganese reducers are almost similar, but a bit lower than that of dehalogenation. Therefore, these electron acceptors compete with each other. They can compete with each other, and they can therefore limit the reductive dehalogenation. Now, this threshold level of hydrogen, it can suggest that in absence of other electron acceptors, dehalogenating organisms should win over methanogens, provided if the hydrogen concentration is below 8 to 10 nanomole. अगर ऐसी सूरत हाल है तो फिर obviously methanogens will remain unable to utilize this hydrogen क्योंकि इसकी concentration कम होगी as compared to the required threshold level concentration. So जो बाकी organisms हैं जो कि dehalogenation में use करेंगे hydrogen को, those can win over and dehalogenation can proceed. This is actually possible more in continuously stirred tank reactors, जहाँ पे continuous stirration होती है, stir continuously करते हैं उसको, वहाँ पे तो possible है, but not in plug flow reactors, because in plug flow reactors concentration increases, hydrogen की concentration बढ़ जाती है, so उसका threshold level ज़्यादा हो जाएगा, methanogens वहाँ prevail करना शुरू कर देंगे. So in order to make sure that methanogens do not prevail, we have to make sure कि जो हाइड्रोजन रिलीज हो रही है by the fermentation of organic matter, उसकी concentration कम से कम हो. Only then dehalogenation can take place. Now, rapid fermentation of organic matter obviously will lead to high concentration of hydrogens, और वो threshold level increase कर जाएगा और उसको फिर methanogens use करना शुरू कर देंगे और वो वहाँ पे grow करेंगे instead of other dehalogenating organisms. The situation can be improved if slowly fermented organic donors are used. अगर हम ऐसे donor use करें जो कि slow fermentation जिनकी होती है, तो hydrogen slowly release होगी, वो फिर उसकी concentration कम रहेगी, threshold level achieve नहीं होगा और methanogens वहाँ पे फिर overgrow नहीं करेंगे. slow release of hydrogen helps to maintain hydrogen near or lower than the hydrogen threshold required for the dehydrogenation. तो इस वजह से जो है जो मेथनोजेंस हैं उनको इतनी कंसंट्रेशन नहीं मिलेगी हाइड्रोजन की कि वो ग्रो कर सकें. However, other microbes will grow and they will start the dehydrogenation process. This was about how different chlorinated aliphatic hydrocarbons can be used as electron acceptors. I will see you in next lessons.